Okay, welcome everyone to the module 3.2 of MOOC on Bioinformatics Essential. And now in this module, we'll learn about running blast and interpreting or validating results of the blast. This will be module 3.2. And before starting with the demonstration of this module, we'll learn about some fundamental theory or fundamental things regarding the blast. Okay, so when we do the blast, what we have to do or what happens during the blast is we take one unknown sequence and we do the sequence similarity search. Then we find the databases matches. We align all the sequence with all possible sequences in the databases, and then we do the alignment and statistics, and then based on the results we get, we do the biological interpretation. So this is the thing, or this is the roadmap of doing blast. Now what is the blast? Blast stands for basic local alignment search tool. So blast is a sequence similarity searching tool developed to identify regions of similarity between biological sequences. So here we do the similarity search between the biological sequences. A query sequence is compared against sequences in a selected database. Okay, so whatever sequence we input, it's called as the query sequence. Okay, so we find the, or we compare that sequence against the selected databases, all the sequences in the selected databases. So it will help to find similar sequence, potential related proteins, conserved regions, possible sequence identity, and candidate functional information. So we can get the functional information of protein, that sequence or protein also. Now important distinction over here is blast primarily identifies sequence similarity. It does not automatically prove biological function or evolutionary homology. So just make sure that, just understand, it does not automatically prove biological function or evolutionary homology. It finds the sequence similarity, and based on that we take the help of blast and other tools like gene ontology and everything to find out the evolutionary homology or biological function of their sequence. So this distinction is extremely important when interpreting the results. Now here, the same reason, the same thing I have highlighted over here, important distinction. This should be understood, and just keep in mind, we just finding the sequence similarity, not proving any biological function or the distinction between the sequences. Okay, now what are the blast programs? What are the different kind of blast we get? So blast 10, where we have the nucleotide in our hand and we have to compare the other nucleotides sequences. So where we have DNA or RNA sequence and it will get compared against the nucleotide sequence. Blast P, so if we have the protein and we have to compare the proteins, used when amino acid sequences compared against protein sequences. Okay, so we give one amino acid sequence and we compare the other amino acid sequences with our sequence. Blast X, it's a translated nucleotide and then protein. So a nucleotide sequence is translated in multiple reading frames, and then we'll compare that against the protein sequences. So if we have the translated nucleotide, then we do the comparison with the protein sequence. And T blastine is a protein, and we do the translated nucleotide. A protein query is compared against translated nucleotide sequences. So we input the protein and we get the translated nucleotide sequences. Now, how to choose the correct program? Okay, so if you have the DNA sequence, we have to start with blastine. If we have the protein sequence, we have to start with blast P. We have the nucleotide sequence but suspect the encoded protein is important, then we got the blastics. We have to do the blastics. And if we have the protein and we want to search against translated nucleotide data, we have to do the T blast N. Okay. More generally, we do the blast N and blast P. Okay, and I will give you the demo regarding the blast N. Okay, blast P is the similar kind of thing. Just you have to paste the thing. Now demonstration session. What we'll do now, we'll meet in the next video for the demonstration of blast, and then I will give the further explanation in and after the demonstration session. Till that point, thank you for the watching the video. um, Give the share, subscribe and like to this video, and we'll meet in the next demonstration video. But make sure you watch all the videos from the module 3.1 part 1 to this video, and then watch the demonstration video, then and then you will get the thing.